so we're going to go through the second section of chapter 12 on neural tissue in this video. And we already talked about neurons, but let's talk about them in depth right now. And so let's go over their characteristics. They are going to be excitable. What does that mean, excitable? They are going to have a polarized membrane. They are going to have differences in charge, like we talked about with the resting membrane potential, where they're going to be more positive outside and more negative inside the cell. That's going to have a graded potential, a charge electrochemical gradient difference from inside and outside. So they are polarized. They are excitable. They live a long time. What you are born with, your brain cells that you were born with are the same ones that you're going to keep your whole life. So long-lived for sure. They are highly metabolic. They need to perform all the time. We can't be deprived of oxygen. They need oxygen. They need sugar. They need glucose as their energy source. They have a high metabolic rate. And so they will die off. You will have brain damage if you have not gotten, oxygen to those brain cells. They can be really large with neurons. If we were talking about specifically ones that go down the leg, some of those are super duper long. And in general, most of your nerves are amitotic. And remember, A means without. So they are going to not undergo mitosis and create new brain cells. Your brain cells are not just, they don't do that in general. CNS are amitotic. They are not going to make new ones. Can they fix damaged ones though? So amitotic, no mitosis. Can they fix damaged ones? Well, some of them can. If you have damaged a nerve, like PNS, if you have damaged one of your peripheral nervous cells, and whatever happened to those cells, we'll talk about that. They can possibly regenerate and fix the damage, but they're still not going to undergo mitosis and make a brand new cell. They will try and fix the damage that has occurred. But if you separate your spinal cord from a terrible traumatic accident, you don't generate all new nerves that run down your spinal cord, but you may be able to fix small damage locally, and maybe we don't fix that, but no mitosis going on. We're not going to completely replace all of these cells like we do with our skin cells. So no characteristics. We always go over characteristics of stuff on the test. So know those parts. And then let's talk about, we talked about parts in general originally, but let's go into details on these. So first we talked about dendrites, and then there were cell bodies, and there's an axon, and that was pretty much all we talked about. This is the picture where we're going to go into details on there. So on the cell bodies are also called soma, and they're going to have some parts. So we're going to have on those cell bodies, they're going to have a nucleus inside of there. And if we're talking about the cells, sometimes these guys will differ on where we consider those. If we cluster all these cell bodies, if we put all of these cell bodies, these somas together, we call them different things in different parts. In the central nervous system, we call all of those nuclei. But in the peripheral nervous system, we call them ganglia. So later when we go into the autonomic nervous system chapter, and you're going to see images of these guys coming off the peripheral nervous system, coming off the spinal cord back there, and you're going to see these chain ganglia, and these groupings, clusters of cell bodies on PNS nerves coming off of those are called ganglia. So PNS, the cell bodies grouped together are ganglia. CNS, the cell bodies grouped together are going to be nuclei. And then inside of there's a whole bunch of Nissl bodies. That's the neuron word for the rough ER. They're called Nissl bodies in here, just like we talked about with muscle cells. They are going to have their own terms. With nerves, they're going to have their own terms too. So the rough ER in a neuron cell is a Nissl body. They're going to have lots of mitochondria, again, because we said they were highly metabolic. And they're also going to have these little guys in here, neurofilaments. Neurofilaments are going to give it structure to that cytoskeleton. So that covers our cell body. Things that lead into the cell body are the dendrites. The dendrites are going to be these guys right here, and all of this stuff, they're off screen. I've covered it up with the other picture, but it should say dendrites. And it's all these guys here, here, here. All of those are dendrites. They are going to have the spines that come down that are going to receive signals. And those signals will end up being graded potentials, and we'll go into further detail on graded potentials in a little bit, that they could be excitatory or they could be inhibitory. But in general, they're going to change their charge, and those are called graded potentials. That happens here at the dendrites that are receiving signals from other cells. So we have talked about dendrites. We have talked about the cell bodies. Let's move on to the axon. And so they're going to have the cell membrane around these things, axolemma, cell membrane on the axon. They're going to have axoplasm, the cytoplasm inside of the axon. They are going to have a special place, and this is one of the most important terms that I want you to know on the whole cell, is the axon hillock. You'll see it called the trigger zone in the chapter outlines, and that is going to be a very important place when those graded potentials that we talk about that come through the dendrites add up to a certain charge that is strong enough to get to this trigger zone, then we trigger an action potential. So that happens at the axon hillock. It is very important, the axon hillock. How many axons does a cell have? All of our neuron cells are only going to have one axon. So even if you see different ones pictured in a little bit, there's still only one axon. It can branch, but there's one. We're going to follow that axon down, and it's going to split as it nears the end of the cell. It's going to split and go into something called telodendria, and then those dead end into these synaptic terminals. You may also see them called axon terminals or terminal bulbs, but that's the very, very end where we have our previously in chapter 10, where we talked about our neuromuscular junction. That is the end of the neuron, and it will synapse with different cells. We're going to cover myelin and its insulating factors again all throughout this chapter. The space in between those glial cells, whether the glial cell is an oligodendrocyte or it is a Schwann cell, the space in between those things are called the nodes of Ranvier. So we need to know all of these terms. Any of these may be possible questions on the upcoming lecture test. When we go down to those axon terminals, synaptic terminals, whatever term you want to use at the end here. So it's coming down, and we are going to have, again, we said these guys are highly metabolic, so there's lots of mitochondria all throughout these cells. They are going to have synaptic vesicles. So before, when we were talking about neuromuscular junction, we talked about how the calcium channel's open, this voltage gated channel pops open, and calcium is allowed to flow in when that action potential comes down the nerve, and it's going to trigger, the calcium coming in is going to trigger that cell to start producing acetylcholine and package it up in vesicles and send it on its way. In this case, we don't know what that neurotransmitter is. It could be acetylcholine, or it could be GABA, or it could be dopamine, or it could be serotonin. There's lots of different neurotransmitters at the very last part of this chapter. So those, whatever that neurotransmitter is, whether it's acetylcholine or any other of the chemicals, they will be packaged up in these things, the synaptic vesicles, and then when it gets an action potential, they're going to send whatever that neurotransmitter chemical is across, and we'll have a chemical ligand signal that is going to go across what we already called the synaptic cleft, the space in between the end of the neuron and whatever it is going to synapse with. And then we have our presynaptic membrane is the end of our original neuron, and then whatever it's synapting with is going to be called postsynaptic, and it could be, again, could be different things. It could be another neuron like we've got here. It could be a skeletal muscle that we've already talked about in our previous chapters, or it could be an endocrine gland when we're talking about hormones. So there's 3 types of synapses when it talks to other cells. That myelin sheath, we said, is important. It is an insulating substance. It is made out of proteins and lipids, and it is the white matter. It's the white matter. So when we talk about gray matter and white matter is what is myelin. Where is the myelin coming from? It's coming from those 2 different cells. And again, Schwann cells in PNS and oligodendrocytes in the CNS. Whatever cell is making it in whatever location it's in, they're both producing the same kind of substance, this insulating factor, this insulating protein lipid that surrounds the axon and helps insulate it, and it gets a better signal that way. So these are myelinated cells. We can see here is our Schwann cell, and it's going to produce the myelin, and you see it just wraps around and around and around and around and around, and it just keeps producing this to wrap that myelin around and insulate the cell. So when we talk about the white matter, again, white matter is insulated. This is the white matter in this picture. The gray matter is non-insulated. So somas, cell bodies, are not insulated, but the axons are insulated. So they would be the white matter, while the cell bodies would be the gray matter. Classifying the neurons. There are different classifications. We can classify them based on their structure, their shape. There is a group of them in previous textbooks, and I don't know if this current textbook mentions it, but they are called anaxonic. And they have lots of dendrites, but no, there's our cell body, our nucleus in there. They don't have a distinguishable axon on it. So these are anaxonic because remember A means that it doesn't have it. So that's one classification where it doesn't have an easily distinguishable axon. All the processes coming off of that thing, all the dendrites in the axon, it looks the same and you can't tell. So we're going to find these in the brain and special sense organs. So in general, would you just say CNS for the brain? Moving on, there are other ones too. So we have unipolars, and these are going to be found in sensory neurons and peripheral nervous. On our sensory, here's our dendrites. They are receiving the information. It is going to go down the axon. And again, there's one axon, and on, here's our dendrites, talking about our bipolars. So on our bipolars, there's going to be dendrites. They send the signal to the cell body. There's the one axon, and it's going to send that information down to the synaptic terminals. Bipolars. Bipolars over here, unipolars over here, one giant axon with the cell body dangling off to the side on unipolars. Where are we going to see unipolars? Sensory, PNS. Where are we going to find bipolars? Eye, specifically in the retina of the eye is where we're going to find bipolars. And then multipolars, we're going to find these on our motor neurons. These are the ones that we've already looked at. Motor, when we talked about stimulation from the nervous system and stimulating the skeletal motors, that's skeletal muscles, that's these guys. These are multipolar. So classified on the shape, do they have no axon where you can't tell what it is? Do they have one giant axon and the cell body is dangling off of the side of it? Do they have a dendritic process that kind of looks like an axon, but it's called a dendritic process, thin cell body, and then the axon flows through like we see in the retina? Or is it a multipolar that to me looks like a tree where we have the branches that meet at the cell body that go down the axon that looks like a trunk and meet at the synaptic terminals at the very end that look like roots for the multipolar? So classifying on shape, anaxonic, bipolars, unipolars, multipolars. Multipolars, handy ones, just telling us again, where are we going to find these guys? These are going to be motor neurons, taking that signal and sending it out for our muscles, for example. Our bipolars, where did we find the bipolars? We found those guys in the retina. And then our unipolars, we found unipolars in other sensory neurons. So you may have a question asking about, this shaped neuron or describe the shape, and maybe it'll tell you is found in the retina. And you need to know, oh, it's talking about the retina, then it's got to be this guy. It's got to be a bipolar neuron. Or maybe it's talking about this type of neuron doesn't have an easily distinguishable axon. Oh, okay. Well, that's anaxonic. So we're definitely going to have some questions on those classifications. There are other classifications too. Does it make a kind of triangular shape? It could be pyramidal. Purkinjes, we're going to have these guys, Purkinjes, in different parts of the body. We have them, here we go, cerebellum, and then olfactory later when we have to memorize the cranial nerves. Olfactory is one of them, taking in that sensory information from smelling things. So cranial nerve, number one, olfactory. And these are the receptors that send that signal up every time you take in a breath and have that air swirl around. Functional classification, yet again, this is another kind of rehash of the diagram that we went into in the previous video, talking about inputs were step number one. Then we needed to integrate it up here as step number two, and then send out an output, step number three, output. So I like these pictures. I think they're really handy. I'm not going to go through it again because we already went through it. But these are great images to help figure out where's the flow, what's coming from what systems are being activated on output. Are we talking about the somatic? We're talking about the autonomic. Where is this information going when we output the stuff? Neuroglial cells is going to be the last part of this video. So we need to know the different types. There are a few. We have mentioned, is it in here? We have mentioned oligodendrocytes, and they produce what substance that insulates things? Myelin, produces myelin. So the oligodendrocytes and the Schwann cells are doing the exact same thing. They're insulators. They are a type of neuroglial cell. They are helpful. There is another type called an astrocyte, and it is sort of, oh my gosh, that was terrible, star-shaped. It is star-shaped. And what does the astrocyte do? It is going to be important for the blood-brain barrier. It's the regulator. It's the bouncer. It decides who gets in and who doesn't. It is going to regulate the ions that get in, the nutrients that get in, the hormones that get in to the brain. It's going to recycle neurotransmitters that are no longer needed. It's the blood-brain barrier bouncer, is an astrocyte. It's an astrocyte. There is this other guy over here, this microglial cell. To me, he kind of looks like a Swiffer. If anybody uses those Swiffer duster things, that's what he looks like. And what he does is it's a macrophage. So it wanders around basically acting like a Swiffer and collecting particles, collecting things that debris, waste products, bad guys, pathogens, and eating them up. He's a phagocyte. So that is a microglial cell. Anything else in this picture? Ooh, ependymals. I forgot about those guys. So the ependymal cells, you can see them at the bottom. And so there's a lot of them and they all line up in a nice little row. And so what these guys do, they are going to produce central spinal fluid. They produce the CSF. So when you get into med micro later, we're going to talk about ways to test and diagnose things. And sometimes patients will come in with decreased neural function, they've got a tightening of the neck, headache, stiffness of the neck. And one of the things that we want to look at is it meningitis? Well, how do we test for meningitis? We got to pull some fluid. We got to pull some spinal fluid out of that area. So who produces the spinal fluid? It's the ependymal cells. They produce the fluid, and then they're going to have cilia on the edge of the cells that help push this fluid around. So it's going to be, we're going to have spinal fluid going around in the brain, and then it goes down into the spinal column and comes back too. So that gets produced by ependymal cells. So oligodendrocytes and Schwann cells are insulating. Microglial cells were Swiffers and they're phagocytes. Astrocytes are the bouncers. They are in charge of the blood-brain barrier. And ependymal cells are going to be in charge of central spinal fluid, the CSF, producing it and swirling it around to get those nutrients that the astrocytes allow to come in. We're going to swirl that stuff around and make sure that we're getting the waste products out, the good stuff in through the spinal fluid. The last cell in here is the satellite cell. And these guys can help with repairing damaged cells. So we said that in general, nervous cells don't undergo mitosis. They're not going to replicate more cells. If they die, those cells die and you don't make more of them. But if they get damaged, there's a possibility that we can fix them, and we fix them with these guys, with satellite cells. Here's our myelin sheath wrapping around, and we said oligodendrocytes and Schwann cells do this. And here's what it looks like. We may have a Schwann cell, and it's wrapping around multiple axons coming through. So we can see all of these different axons in this one Schwann cell, and it's wrapped it up in a pretty nice, tight little bundle, insulating all of those axons. What happens if it gets damaged? Can you fix it? Yes, you can sometimes fix it if conditions are right, and the injury meets, the injury is meeting these qualifications, and there's a possibility that that injury may be repaired. So what are our steps if something happens? So we have damaged this neuron here. Here's our site of injury. So I'm going to use myself as an example. I grew up doing dive team when I was younger, and I did the low board and the high board, and nobody has high dives anymore, but our club had a high dive and back in the day. And so I was doing a dive off the high dive, and I was doing something called an inward, and that's where you stand backwards on the board, you bounce off, and you do your moves towards the board as you fall towards the ground. So I was doing an inward somersault, and so I bounced off, and I went to do my flip, and I tucked really tight, but I didn't bounce out far enough, and I cracked my head on the diving board, and so it split right across. It was a bloody mess, and it was super embarrassing, and then the lifeguard came and got me out of the pool. Really embarrassing story, especially because I thought it was kind of cute. Oh, it was terrible. Anyway, they fished me out. It sliced right across. You could see my skull. It was terrible. And so I did not have any sensation after they stitched me up. I had like 12 stitches. I couldn't feel anything around the damage. It was numb. I was brushing my hair and I didn't feel it because of that injury. But luckily, my injury was like this. So in step one, we have the damage, right? And so we've got a bit over here, we've got a sump over here, and then we've got our axon over here, and then we've got all these pieces, these fragments, these pieces of junk that we need to get rid of. So Schwann cells are here, and then we're going to have macrophages come in. So whether it's a microglial cell or some other type of macrophage, but they're going to come in and remove the damaged bits, all the broken bits, and we'll take those and clean it up and make it better. So that's step two. We are going to stimulate the axon to continue to grow and try and go into that damaged area while we clear out the damage with a macrophage. And so step three, the axon's going to keep on growing in the direction that it was already going in the first place. So it just keeps going in the same direction. And if that same direction is still where the other side of the neuron is, so it can meet up, because the damage, it busted up right here in the middle, but they were able to grow together. The alignment was okay. It wasn't misaligned and they missed each other. They were still aligned And so they were able to grow back together. So it can repair itself if conditions are right and everything can get back in alignment. There's a possibility of repair. So I had my injury and they stitched it all up. And for a very long time, I could not feel that. And I just figured that that's how it was going to be my whole life. It would just be numb up here, but I can. It finally did. It grew back. So it can repair itself. Peripheral nerves can repair themselves. Just takes time. So it takes time and it has to have the proper conditions. So that finishes out this video. The next video, we're going to combine the next two sections and talk about graded potentials and action potentials and how that stuff happens.